Hello once again. Welcome to Finals Week 3 lecture session. So for today, our topic are non-fermentative gram-negative bacilli. So the learning objectives for this topic would be, at the end of this unit or this discussion, the student should be able to characterize the significant species of non-fermenting gram-negative bacilli, their general properties, and virulence factors, to describe and explain the clinical significance and pathogenesis of the diseases caused by these organisms, and to distinguish features of non-fermenters for accurate laboratory diagnosis and determine possible treatments. Okay, so for the lineup of topics, we will be discussing the NF-GNB. Okay, so these are able to metabolize carbohydrates oxidatively and would require molecular oxygen as final hydrogen acceptor. Okay, so we use the term non-fermenters because they cannot ferment glucose or other carbohydrates in the absence of oxygen. Okay, so the oxidative process usually results in the formation of very weak acids as end products of their metabolism, unlike those of the members of Enterobacteriaceae family. Okay, we will also be discussing other gram-negative bacilli, such as the non-saccharolytic or non-oxidizers, those that do not utilize carbohydrates with or without oxygen, and they usually make use of other compounds as source of energy, like, for example, amino acids. Okay. So here's a short flow chart to show you how we could easily differentiate gram-negative bacilli. So since this is a large group of organisms, okay, mainly populated by Enterobacteriaceae, okay, so we can easily differentiate them by performing oxidase test. Okay, so oxidase negative organisms, okay, can be tested further with the OF medium. So they are positive for oxidizing carbohydrates as well as fermenting carbohydrates. Okay? Largely, organisms with this type of reactions are Enterobacteriaceae. So oxidase test negative, but they're able to demonstrate positive OF reactions on Hugh-Lawson's media. Okay, now oxidase positive organisms may be tested also using the OF media. Those that show a variety or variable reactions to oxidation and fermentation using the OF media might be Pseudomonads or Vibrios. Okay? Pseudomonads are positive for oxidation, but they cannot ferment carbohydrates. Okay? Vibrios, on the other hand, are positive for both oxidation and fermentation. So they are similar. The reaction to OF in OF media is similar to Enterobacteriaceae, but they are, of course, oxidase positive. Okay. So to characterize non-fermenters, okay, so they are non-spore formers. They are obligate aerobes. On TSI, they usually demonstrate red slant, red butt, or alkaline slant, alkaline butt, and no hydrogen sulfide production. A point of colonization of non-fermenters are usually the mucous membrane as well as the skin of humans. Okay, so they can enter sterile sites in the body via trauma, burns, or wounds. Okay, a large number of these organisms are opportunistic pathogens. Okay, so a non-fermenter is suspected if the reactions to the laboratory test come up as oxidase positive gram-negative bacilli. They grow better in sheep's blood agar than on McConkey agar. Okay, and on TSI they show red slant, red butt. Okay, now to further differentiate or identify rather non-fermenters, you can perform, of course, you can check for flagellar morphology. A lot of these are motile. Okay, so you can check out the arrangement of their flagella, the number and arrangement of their flagella. Of course, you can perform indole test, but for indole test in identifying non-fermenters, it has to be modified. Okay, why? Because non-fermenters produce only a small amount of indole. Therefore, indole needs to be extracted first so that it could be detected. Okay, so in the same manner, you'll be using SIM, no, or tryptophan broth. Okay, and then the organism is inoculated in it, and it is incubated usually until 48 hours at 25 to 37 degrees Celsius. Okay. Now, the modification usually comes in after inoculation and incubation. Okay. So the next day or on the 48 hour of incubation, you add about 1 ml of extractor like or extractant like xylene or chloroform. Okay. So you add it to your SIM. Okay. And then, or if you're going to use a tryptophan broth, so you add it and then you shake it to mix and then let it settle for some time until the solvent rises to the surface of the broth. Okay. And then that's the time to add PDAB. Okay. But here for the modified indole, we'll be using Ehrlich's reagent instead of the Kovacs. Okay, so basically they are similar. Now they're the same. They contain PDAB, but Ehrlich's contains ethyl alcohol. No, an addition of ethyl alcohol. Okay, so it is said to be more sensitive, no, than Kovacs when detecting indole produced by non-fermenters. Okay, the reaction, the positive reaction is still the same. Now you'll be looking for that red ring at the top of the broth. Okay, then you can also perform growth on McConkey. Okay, and then motility test. Again, the OF test. Okay, this is very important for non-fermenters. Okay, so we also use this not only for Enterobacteriaceae, but we also use this for non-fermenters. Okay, so we check their OF carbohydrate utilization patterns. We can also perform nitrate reduction tests. That these two tests will be discussed shortly. Okay, then we can perform acetamide. Okay, so at the acetamide utilization test. Okay, this is similar to citrate test. No? So it is a test to check the ability of the organism to use acetamide as a source of carbon. So basically, the reaction is similar. Okay, so if the organism was able to utilize acetamide in the agar, okay, that would turn the agar blue. Okay, so negative reaction green. Okay, we can also check for urease production and the carboxylase reactions. Okay, so as promised, we'll be discussing the OF test using the Hugh and Leifson's media. Okay, so the OF medium has been formulated to for the study of carbohydrate utilization for non-fermenters or by non-fermenters. Okay, so this media has a high concentration of carbohydrate, which is 1%. That's high already, kids. Okay, so high concentration of carbohydrates at 1% and low concentration of peptone or protein at 0.2%. So see, one is already high because the peptone is low at 0.2. Okay, so the high concentration of carbohydrate enhances the production of acidic end products, which can be detected. Because remember, our non-fermenters are kuripot, no, in producing acid. So we need a higher concentration of carbohydrates so they will be able to use it more and therefore produce acids more. Okay. So that's the purpose of the high carbohydrate concentration. Now, the low concentration of peptone, in this case, 0.2%. Technically, in most culture media, it's around 2%. So here, the concentration is 0.2%. Why? Okay. The low concentration of peptone decreases the formulation of oxidative products and amino acids. Kasi these products can neutralize the pH, and therefore, if the pH is neutralized, it will obscure the acid, or it will mask the acid that has been produced previously. Okay, so mawala, it will not be red anymore. Okay. So the concentration of peptone to carbohydrate is 1:5 in OF medium for non-fermenters. Okay, so 1, that's 0.2, and then 5, yung 1%. No? So concentration of peptone, or ratio of peptone to carbohydrate concentration in OF medium for non-fermenters is 1:5. Whereas the concentration of peptone to carbohydrates in OF medium for Enterobacteriaceae is around 2:1. So the concentration of peptone is higher than the carbohydrate concentration. Okay, so that's for Enterobacteriaceae. That's the main difference for the OF that's used for non-fermenters and the OF used for Enterobacteriaceae. Okay. So why can't we use the 2:1 concentration for non-fermenters? Again, no? So the amount of acid produced by our non-fermenters is not sufficient, no, to trigger the indicator included, no, to change the color of the medium from green to yellow. Kasi cute lang yung amount. Okay. Secondly, the alkaline end products produced during the reaction will neutralize the acid produced. Konting-konti na nga lang yung acid na na-produce. Okay? Natakpan pa nung andaming alkaline products. Okay? So, ang kinalabasan niyan, all the reactions will be false neg, if you're going to use the 2:1 concentration. That's why if you're going to identify non-fermenters, we have to stick with the 1:5 concentration of peptone to carbohydrates. Okay, so again, the concentration of carbohydrates is higher, okay, than the concentration of peptone for the OF na gagamitin to identify non-fermenters. Okay. So this is the usual reaction. So we will be able to identify 3 results, okay, or 3 reactions for oxidative fermentation using the Hewen-Leifson's media. Okay, so the tubes are in pairs. Okay, so first pair, second pair, third pair. Okay, so for the first pair, okay, to the left is the open tube, and to the right is the closed tube. It's closed because a sterile mineral oil or sealant, okay, it could be petrolatum, or in this case, mineral oil, is added at the top of the solution, or the media rather, to seal, okay, to seal the tube. Okay, so for the open tube, we only have cotton plug, no, or not so tightly closed screw cap. Okay, so on the first pair, or in the first pair, you observe that in the open tube, the culture media remained green. No? So the original color of the OF media is green, emerald green. Okay, so this means no carbohydrate has been utilized, therefore no acid has been produced. Hence, no color, no yellow color produced. On the closed tube to the right, the color is still green. Okay, in some cases it might be blue-green. Okay, so they're technically the same, green or blue-green. So in the second tube, no, the closed tube, it's still green. Okay, so if we define this, the first tube is for oxidation. Okay, the second tube is for fermentation. Okay, so in this case, the organism inoculated is not able to oxidize nor ferment carbohydrates. So they are non-saccharolytic or non-oxidative. Okay, they're not able to utilize carbohydrates, okay, as their source of energy. in the second pair of tube, notice that in the open tube, there's already a yellow color, meaning carbohydrates has been utilized. Therefore, it has changed. It has produced acids that has caused the change in the color of the medium from green to yellow. Okay. In the second tube, the closed tube, it remained green. Okay. So this means that they were not able to ferment carbohydrates in this tube, but they were able to oxidize carbohydrates in this tube. Hence, the result is reported as oxidative. In the third pair of tubes, notice that both open and closed tubes have turned completely yellow. So these are organisms that are strongly able to oxidize carbohydrates and ferment carbohydrates. Okay, so they're called fermentative organisms. Okay, so pag green ang duha, open and closed, non-saccharolytic or non-oxidative. Kung positive sa open, negative sa closed, oxidative. Okay, kung yellow in both open and closed tubes, fermentative. Okay. So the first set of tubes is characteristic of Alcaligenes faecalis, whereas the second pair is characteristic of Pseudomonas, and the third pair is characteristic of Enterobacteriaceae. Okay. Oh, may ganun-ganun pa. Now, moving on to nitrate reduction test. Okay. Asan na naulihi? Labasin na lang muna natin silang lahat. May ganun. Ayan. Okay, so gusto ko nakalabas na silang lahat. Okay, so the nitrate reduction test, as previously mentioned, is another test that can be used to identify non-fermenters. Okay? So nitrate broth is usually used to demonstrate the ability of the organism to reduce nitrate to nitrite. So in the nitrate broth, if the organism possesses or is able to produce the nitrate reductase, it will reduce the nitrate in the broth into nitrite. And sometimes it can be further reduced into nitrogen gas, nitrous oxide, or nitric oxide. Okay. Now to verify, okay, we add sulfonylic acid and alpha-naphthylamine. Okay. Now, if a red color develops, that means a red diazonium salt has been produced, which indicates a positive result for nitrate reduction to nitrite. However, there are cases wherein there is no red color production. Okay, so there's no red color production. Why is that so? Okay, so sometimes this happens, okay, because number one, the nitrate may not have been reduced, which means that the organism that has been inoculated is nitrate reduction negative. No, it's not able to reduce nitrate to nitrite. Or it could be that the organism inoculated is able to reduce nitrate to nitrite and has further reduced the nitrite into gas. Okay, so therefore, sulfonylic acid and alpha-naphthylamide has nothing to identify because it has already turned into gas. Okay, so to verify this, we can add zinc dust. Okay, so when zinc dust is added, okay, we have to wait for the appearance of red color. Okay, now if no red color develops, that means nitrate has been reduced to nitrite. Why so? Zinc dust will identify nitrate. Okay, and since there's no red color production, it means that the nitrate indeed has been reduced into something else. Okay. However, if a red color develops, that means zinc has identified nitrate, which means that nitrate has not been reduced. So it's negative. It's a negative result. Okay, so no red color, positive for nitrate reduction. With red color, negative for nitrate reduction. Okay, again, okay, let me show you the procedure. Okay, so nitrate broth that's inoculated with an organism will be incubated at 37 degrees Celsius for 24 hours or up to 48 hours. Okay. Then we add 10 drops sulfonilic acid, 10 drops alpha-naphthylamine, and observe for the production of red color. Okay. Iron. Ayan. Okay. So if red color has developed, that signifies reduction of nitrate to nitrite. However, okay, if it has not changed its color, no red color production, we will have to verify it, okay, because this reaction may mean two things. It could mean that the nitrate has not been reduced because non-reducer ang organism, or it could be that it has been reduced further into gas. Nitrite has been reduced further into gas. So the added sulfonilic acid and alpha-naphthylamine has nothing to identify. Okay, that's why they were not able to produce the red color. So to verify, we have to add zinc dust. And remember that zinc dust will identify nitrate. Okay, so upon the addition of zinc dust, if a red color develops, that means the nitrate has not been reduced. However, if no red color develops, that means the nitrate in the culture media has been reduced. Okay, therefore, zinc will not have anything to identify. Okay, so no red color on this portion is positive for nitrate reduction. Red color in this portion means positive for nitrate reduction. Okay, so you only do this portion to verify if this reaction comes out. Okay, so these are actual pictures. So no change upon addition of alpha-naphthylamine and sulfonilic acid, meaning nitrite is absent. So this tube here will require the addition of zinc dust. Okay, this one is a positive result already. So after the addition of alpha-naphthylamine and sulfonilic acid, it has turned red. So that's a go. That's already positive. Okay, now after adding zinc dust in this portion and no change has happened after adding zinc, so it's a positive reaction for zinc reduction. However, after adding zinc here and red color has been produced, it's a negative reaction for nitrate reduction. Okay, claro, no? So let's start with our first group of organisms. So we have our family Pseudomonaceae. Okay, so the non-fermenting gram-negative bacilli. Okay, so there are 5 groups in this family. Okay, so the first group are the fluorescent group of organisms, which includes Pseudomonas aeruginosa. Okay, this species can produce several pigments, including pyoverdin and pyocyanin. Other members of the fluorescent group includes Pseudomonas fluorescens and Pseudomonas putida. Both can produce pyoverdin only. Then we also have non-fluorescent members in group 1. This includes Pseudomonas tetzeri and Pseudomonas mendocina. Okay? Both are soil denitrifiers. Okay? The second group are the Pseudomallei group. These are said to be resistant to polymyxin B and colistin. It includes Burkholderia pseudomallei, Burkholderia mallei, Burkholderia cepacia, and Ralstonia piqueti. Now, Burkholderia pseudomallei, Mallei, cepacia, and Stenotrophomonas maltophilia at the end are all able to again resist polymyxin B and colistin. Okay, they are actually considered the pseudomallei group, including this organism. Okay, Ralstonia piqueti, Comamona species, and Acidoborax species, as well as the Brevundimona species, are have not been isolated as much as these organisms in human infection. So they're technically really rare, okay, and might have been isolated from the environment, okay. So let's start with the first clinically significant species of Pseudomonas, and that's Pseudomonas aeruginosa, okay, or PAE. This is the most frequently isolated non-fermenter. Okay, it's not a major normal flora, but it's the most frequently isolated non-fermenter. Okay, Pseudomonas aeruginosa is nicknamed PAE, no, PAE. Okay, so this accounts for about 75 to 80% of non-fermenting gram-negative bacteria isolated from clinical specimen. Okay, it is mostly an opportunistic pathogen. Okay, it can be identified by virtue of the pigments it produces. No, it can produce pyocyanin or the blue pigment, pyoverdin or the green pigment. Sometimes they can produce the fluorescent pigment, yellow-green color. Okay. Others can produce brown, red pigment. Okay. So these are organisms that are mostly found in the environment. They can survive in harsh environments. Okay. Among the Pseudomonas group, this is considered as a major human pathogen. Okay. It is invasive and quite toxigenic. That's why it's considered as the most important opportunistic pathogen. Okay. So characteristics of our Pseudomonas aeruginosa are as follows. It's an obligate aerobic organism. It's motile by virtue of a polar flagellum. Again, it produces the pigment pyocyanin, pyoverdin, fluorescein. Okay, so this is pyoverdin. This is fluorescein, so neon. This is pyocyanin, pyomelanin. This is a combination actually, pyocyanin and fluorescein. Okay? This is also a combination, pyoverdin and fluorescein. This is pyorubrin or the red pigment. Okay. The last one shows no pigmentation. Okay. Now, Pseudomonas aeruginosa is able to produce 2-aminoacetophenone, which allows it to release a fruity odor or corn tortilla-like odor, which is characteristic for Pseudomonas aeruginosa. So mabango, no? Amoy pagkain. Okay, so it grows at different temperatures. It can grow at room temperature, it can grow at 35 to 37 degrees Celsius, and it can even grow at 42 degrees Celsius. Okay. So this ability to grow at this temperature separates it from the other two fluorescent species of Pseudomonas, Putida and fluorescens. Okay, because those two cannot grow at 42 degrees Celsius. Okay, so going to laboratory tests. Okay, so specimen best cultured or processed for identification of Pseudomonas includes skin lesions, pus. Okay, pus would be best. Urine, blood, spinal fluid, sputum, and others. Okay, in gram staining, there are of course gram-negative rods. When cultured on blood agar plate, they can produce large, rough, grayish colonies like this, and their edges are serrated, no, giring-giring ang edges. Okay, mostly they are beta hemolytic. Okay, sometimes some strains can produce capsules with alginate, so the appearance of the colonies are mucoidal, okay, or mucoid. Okay. On MAC and EMB, they are non-lactose fermenters. Now, biochemical tests, on TSI, they can produce alkaline slant over alkaline butt, oxidase positive, oxidative fermentation. They are able to ferment and oxidize glucose, fructose, xylose, but not maltose, sucrose, and lactose. Okay. And they're also able to reduce nitrate to nitrite. So this is a table showing a summary of the different lab tests that you can check out to identify Pseudomonas aeruginosa. So those highlighted are already identified. Okay, so you can check out those other not highlighted tests, to further differentiate them. Okay, so for the infections associated with Pseudomonas aeruginosa, they are, of course, associated with a lot of nosocomial or hospital-acquired infections, okay? Like severe wound infections in patients with burned skin like this, no? Ayan, so exposed, wala yung first line of defense kaya nasunog. Okay, so it's like adding insult to injury. Okay, it can also cause UTI. It can cause pneumonia, especially ventilator-associated pneumonia. Okay, septicemia, chronic lung infections in patients with cystic fibrosis, especially the mucoid strain. It is also known to produce meningitis and an infection called ecthyma gangrenosum. Okay, so ecthyma gangrenosum, this one, is a cutaneous infection associated with Pseudomonas bacteremia. Okay, so it usually occurs in patients who are critically ill or bedridden and immune compromised. Okay, so the characteristic lesions of ecthyma are hemorrhagic like this. So it usually starts out like this. Now there's a small infected area, but the surrounding area is already inflamed. No? It mimics cellulitis. Eventually, it will develop macular rash and a papular rash that will grow in size and contain blood. No? So the pustules are bloody or hemorrhagic. And then later on, okay, as it pops, it creates that necrotic ulcer or blackened center. Okay, so we can observe this usually in bed sores like that. No? So these are the most common hospital-acquired infections associated with Pseudomonas. Now, Pseudomonas is also associated with a bunch of community-acquired infections, no, like keratitis, no, like this. Okay? So inflammation of the cornea. Then we have corneal ulcers, especially those wearing contact lenses. So you have to be very careful with your eyes, especially when you're wearing contact lenses. I had a friend once who really needed contact lenses. She uses it kasi doble vista, ang kapal ng salamin. So minsan, the weight of the eyeglasses bears down onto the nose, no? The bridge of her nose, masakit daw. So that's the time she uses her contact lenses. And at one time, she forgot to take it off before going to bed. The next day, her eyes looked like this. And nagka-scar yung kanyang cornea. But this is actually treatable. Okay? As long as it has been diagnosed early and it is treated with the proper antibiotic treatment or therapy, and the treatment is religiously followed by the patient, it's actually treatable. Okay, so swimmer's ear is also associated with Pseudomonas aeruginosa. No? In our dialect, we call this buog, buogon. And then folliculitis, no? The hot tub folliculitis type. Usually this is acquired in bathing in warm tubs that is contaminated with the bacteria. Okay, nakikiswimming rin si Pseudomonas. Okay, so Pseudomonas are able to cause those infections because of the virulence factors. Okay, so these are the virulence factors that contributes to its pathogenicity. It includes the structures for attachment, such as fimbriae. Okay, so fimbriae, it allows attachment to respiratory epithelium. It can also activate pro-inflammatory genes. Okay, flagella, no, it is motile because of a polar flagellum. It is, apart from the organ of motility, it also enables the organism to adhere to host cells and activate interleukin 8. Okay, interleukin 8 Maybe you have discussed this in your serology subject. It's an important protein related to inflammation. It plays a key role in recruitment of neutrophils and other immune cells to the site of infection. Okay, and then virulence factors also include endotoxins and exotoxins, no, like lipopolysaccharide endotoxins, no, such as the alginate capsules, or exotoxins such as exotoxin A, which inhibits protein synthesis and facilitate infection. This is actually the virulence factor largely responsible for tissue necrosis that we see in ecthyma. Okay, and then type 3 secretory system, or secretion system. This injects toxins to host cells, okay? And those toxins would have cytotoxic effects. Okay? And then they have exotoxin S, T, U, and Y, which also has cytotoxic effects. And then, of course, for tissue invasion, okay, they are able to produce elastase, which digests elastin, protease, which degrades protein, collagenase, which destroys collagen, and of course, hemolysin and leucocidin, which destroys blood cells. And their pigments, no, pyocyanin and pyoverdin. Okay? Pyocyanin, for one, is known to inhibit lymphocytes and cilia. And pyoverdin binds iron. Okay. So for treatment, treatment of Pseudomonas would require combination treatment. No? Single drug therapy is not recommended because in single drug therapy, the success rate is so slow. And also, the bacteria has the capacity to develop resistance. Okay, so usually it's a combination between a penicillin-based drug and an aminoglycoside. Okay, so beta-lactam drugs can also be used. Third and fourth generation cephalosporins like ticarcillin and piperacillin can be used. Okay. Carbapenems, these are also beta-lactam drugs. This has the broadest spectrum, and more effective than other beta-lactam antibiotics against Pseudomonas. They're also synergistic with aminoglycosides, so you can pair that. Okay, and then for fluoroquinolones, it can be used to inhibit nucleic acid synthesis. So you have choices such as ciprofloxacin, levofloxacin, or ofloxacin. Okay. Again, it's important to perform antimicrobial susceptibility testing to be sure. Okay, so you have to wait for the result of AST before treatment has to be handed out. Okay, so moving on to the remaining fluorescent species of Pseudomonas, we have fluorescens and putida. Okay, so again, these are fluorescent pseudomonads. Okay, they can produce pyoverdin, but they cannot produce pyocyanin. Only Pseudomonas aeruginosa can produce that. So they are found in the environment. Okay, they can grow at 4 degrees Celsius and cause transfusion-associated sepsis. Also, it's associated with nosocomial infections. Okay, it's not part of the normal flora, but they can be associated with opportunistic infections, but not as common as Pseudomonas aeruginosa. Okay, see, fluorescence is a contaminant. It can cause food spoilage or sometimes plant infections. It can cause UTI, and wound infections in humans, but at a very rare, for very rare cases. Okay? Putida has been seen in isolated cases of septicemia, UTI, and pneumonia. And it has been isolated in the hospital environment, especially around sinks, okay, sa lababo. Okay, so the laboratory characteristics of Pseudomonas, fluorescens, and putida are as follows. No? So they are motile, both by virtue of a Lophotricus flagella. Okay, they grow at 35 degrees Celsius, but cannot grow at 42 degrees Celsius. So that's another differentiating point from Pseudomonas aeruginosa. They're oxidase positive, of course. OF medium, they can oxidize glucose only. Okay, pyocyanin negative, pyoverdin positive. For citramide and acetamide, they are negative. For arginine, they are positive. They're resistant to carbonicillin. Okay, and then they are susceptible to kanamycin. Okay, so to differentiate the two, since they are closely similar in their laboratory characteristics, we can perform protein metabolism and gelatin hydrolysis. For protein metabolism, fluorescence is proteolytic, whereas putida is non-proteolytic. For gelatin hydrolysis, fluorescence is positive, putida is negative. Okay. Now moving on to the non-fluorescent group, which are Sitzeri and Mendocina. These are soil denitrifiers because this bacteria use ammonium as a sole nitrogen source and acetate as their sole carbon source. Okay? They can grow anaerobically, they can produce nitrogen gas. Sometimes they have been isolated from straw or the yami, and in manure, animal manure, sometimes in contaminated cosmetics, ayan, okay? And even in baby formula, okay? They can be rare causes of eye infections, endocarditis, septic arthritis, post-surgical wound infections, UTI, septicemia, and pneumonia. They are motile by virtue of a polar flagellum, no? So monotricous ito sila. Okay? They can grow also at 42 degrees Celsius. Okay? Sitzeri is usually found in soil, no, in marine water and medical devices. Its colonies, when cultivated, appear dry, ayan, and wrinkled with buff or light brown color. Okay? For arginine dihydrolase, they are negative, but they are positive for starch hydrolysis. Okay? On the other hand, Mendocina can produce smooth, no, buttery, flat, and wrinkled colonies on blood agar plate. And they are positive for ADH and negative for starch hydrolysis. So observe, opposite yung reaction nila sa dalawang test na to. Okay, so that's how we differentiate the two members of the non-fluorescent group. Okay, so moving on with the next group, okay, still belonging to the family Pseudomonadaceae. So we have the Burkholderia group. Previously, they are part or members of the Pseudomonas group. Okay, so the genus, no, was named after Walter Burkholder, a plant pathologist. He was able to first describe Pseudomonas cepacia, which is later on called or labeled as Burkholderia instead of Pseudomonas. So we have Burkholderia cepacia. Okay, so these are commonly seen in the environment. No? So marami sila. These are ubiquitous gram-negative bacilli. They are obligate aerobes. They are motile by virtue of a single or sometimes Lophotricus. Okay? So single or Lophotricus flagella, except Malay, because Malay is non-motile. Okay, so there are 4 known pathogenic species of Burkholderia. We have Burkholderia malay, which is the agent of Glander's disease, technically a zoonotic disease. We have Pseudomalay, which can produce a Glander's-like disease called melioidosis, and then Cepacia complex, which can also cause infections in humans, and Cepacia, which is associated with pulmonary infections in patients with cystic fibrosis. Okay, first species. We have Burkholderia malay. Okay, so Burkholderia male is the causative agent of glanders disease. It's again primarily a zoonotic disease. No? It affects horses. However, since we do associate with horses a lot, okay, so the disease can be transmitted to humans, but the transmission is from the infected horse to humans. Human to human transmission is really rare. Okay, so if this happens, the transmission is usually through direct contact via skin abrasions or inhalation or contact with the mucous membranes of the eyes and the nose. Okay, now for laboratory diagnosis, male again is non-motile. It is resistant to polymyxin B. It is weakly oxidase positive. It cannot grow at 42 degrees Celsius. And it can produce smooth cream to white colonies on BAP or blood agar plate. Second species, we have Pseudomalae. Okay, so Burkholderia pseudomalae is a causative agent of melioidosis, which is a glanders-like disease, or also known as Whitmore's disease. Okay, so melioidosis is an infectious disease. You know? It can affect people, but some experience no symptoms. No, others may experience symptoms, and the signs may be quite mild, like fever, skin changes. Some may experience pneumonia and abscesses. Okay? Sometimes the symptoms can be severe, like inflammation of the brain, inflammation of the joints. Some may experience dangerously low blood pressure, which may cause multiple organ failure and hence end with death. Okay. So it may be an aggressive granulomatous pulmonary disease for some. Some individuals may be asymptomatic. Okay, so lab diagnosis for Burkholderia pseudomalae, we can grow them on sheep's blood agar. They produce wrinkled cream to tan colored colonies. We can also grow them on ashdown medium. There, they produce deep pink colonies with a characteristic earthy amoy lupa, earthy odor. Okay? The components include crystal violet and gentamicin, making the ashdown medium a selective culture media. Okay, so the selective agents are is gentamicin. Okay, so it suppresses unwanted bacteria. Okay, there's also 4% glycerol, which is required by some strains, and then neutral red. This is responsible for the deep pink color produced by the colonies. Okay. Now, Burkholderia pseudomalae is oxidase positive. It's motile. It's a Lophotricus bacteria. It is arginine positive. And for OF medium, it can oxidize glucose, maltose, lactose, and cellulobiose. Okay. Then we have Burkholderia cepacia naman. It is said to be the agent of foot rot in humans as well as animals as well. Okay, even in roots of onion bulbs. Okay, so it can cause pagkabulok. Okay, so very common in individuals who are who are usually using boots or rubber shoes with non-absorbent socks. Ayan. So this is also known as trench foot. Okay, so the first cases of this were observed in American soldiers several decades ago. They were fighting wars, and their boots are usually immersed in water or in marshes, marshes rather. And so, ayan, nabulok. So, nakulong. This one, he had his boots on for 10 hours. Okay, so, nalata yung tiil. Kasi basa ang boots. Okay, ito naman, naka-rubber shoes ng matagal. Okay. So, ayan. Okay, so this organism is actually an opportunistic bacteria as well as a nosocomial organism. Okay. It's usually found in soil, in water, in plants. Okay? They can very well survive in the hospital environment. Okay? It can also, apart from foot rot, they can also cause pneumonia, especially in patients with cystic fibrosis. Okay? They can cause UTI, septicemia, endocarditis. It's not a normal flora of humans, and it's not pathogenic in healthy hosts. Okay? So laboratory diagnosis, so we can check out their motility, their motile with polar flagella. Their colonies are yellow colored. The edges are serrated. Okay, they are weakly oxidase positive. Okay, in OF medium, they can oxidize glucose, maltose, lactose, and mannitol. Okay, for treatment, okay, we can use sulfamethoxazole, chloramphenicol, and other third generation cephalosporins. Okay, wrong spelling pa. Okay, next we have Stenotrophomonas maltophilia. Okay, so these are found in food and water sources, in soil, in animal as well as plants. Okay, they can also be very well isolated in clinical sites in the human body. Okay, they are contaminants or commensals on humans. Okay, they can be transient flora. Okay, it can be acquired in the hospitals. They can colonize the respiratory tract, the genitourinary tract. They can cause nosocomial infections. No? They can also cause pneumonia, septicemia, UTI, pare-pareho lang, di ba? Oh, and meningitis. Okay? So laboratory diagnosis, okay, for Stenotrophomonas, they are of course motile with polar flagella. They produce large, smooth, glistening colonies with a yellow to tan pigment on TSA. Okay, they cannot produce pigmentations like pyocyanin or pigments rather. They cannot produce pigments like pyocyanin or pyoverdin. Okay, and then they can produce lavender green to light purple pigmented colonies on sheep's blood agar. They can also grow at 42 degrees Celsius. Okay? What's unique about them is that they are oxidase negative. Sa lahat naman ng kasama niya, siya lang yung oxidase negative. Okay? So in OF medium, they are weak oxidizers of glucose but very, very strong oxidizers of maltose. That's why they're called maltophilia, which means maltose loving. Okay, they are also oxidase positive, okay, but they cannot reduce nitrate to nitrite. Their colonies may have a characteristic ammonia-like odor. Okay, so for treatment, they are still treatable with trimethoprim sulfamethoxazole. Okay, so here is a summary of the different biochemical reactions that you can review and the different species for non-fermenters. Okay, so simula kay Pseudomonas aeruginosa hanggang kay Stenotrophomonas. Okay, so you can use this to help you review. Now let's move on to Acinetobacter. Okay, so Acinetobacter are nosocomial and opportunistic pathogens. They can colonize moist areas of the human body. Ayan. So they are able to cause infections like UTI, respiratory tract infections, wound infections, bacteremia, and meningitis. Okay? Previously, they were called MIMA. Okay, like Mima polymorpha, because they're able to mimic the appearance of Neisseria. That's why, to be sure, we have to perform we have to perform oxidase test, okay, to do this. Because our Acinetobacter is oxidase negative, while our Neisseria is oxidase positive. Now, there are two common human pathogens. We have Acinetobacter baumannii and Acinetobacter loofii. Diba? Loofii and baumannii. Laboratory diagnosis to identify Acinetobacter, they are obligate aerobes, again, oxidase negative, catalase positive, and they are non-motile. Okay. Unlike those previously discussed, this one is Coco Bacilli, so rather short and stout. Okay. Busok. Okay. They grow on EMB and MAC. Okay. They are non-hemolytic on sheep's blood agar. Okay. And they are, of course, resistant still to penicillin, aplinabilin. Okay. So to differentiate baumannii and loofii, so we have this. Okay. So both are, of course, oxidase negative, lysine decarboxylase negative. So the test that you can use to differentiate them is growth at 42 degrees Celsius. Baumannii is positive. For glucose, baumannii can ferment it rapidly, whereas loofii is a saccharolytic. Okay. For lactose assimilation, baumannii can rapidly assimilate lactose, and it is positive for fluorescence lactose denitrification acid test. Okay? Whereas loofii is negative. So ito naman yung mega-nega species. Okay? Si loofii. Okay? Then we have Alcaligenes. Okay? So Alcaligenes is a tiny gram-negative bacilli. It's motile because of Peritricus flagella. Okay? Clinically significant species is marami, but the most commonly isolated is Faecalis. Okay? So Faecalis was previously called odorans, no? Alcaligenes odorans. But now they retain Faecalis. Okay? So it has a characteristic fruity odor of apples or pears. So kahit na maliit lang yung number ng colonies, ang lakas ng amoy daw ng fruity odor ng apples. Okay? It is able to cause several opportunistic infections. A laboratory diagnosis, it's oxidase and catalase positive, and it can grow on MAC conchi. On sheep's blood agar, it is able to produce flat, dull colonies with irregular margins. Okay, so beside that is a table to show you the difference between Faecalis and another species. Okay, so maraming species, but Faecalis is the most commonly isolated. Okay, now key points before we move on to the next group of organisms. The key points for the first group are the following. Okay, so the most common non-fermenters isolated in clinical microbiology laboratories are Pseudomonas aeruginosa, Acinetobacter baumannii, a species, a complex, Stenotrophomonas, Maltophilia, and Burkholderia cepacia complex. Okay, most non-fermenters are oxidase positive. Okay, it's a key test in differentiating it from most members of the Enterobacteriaceae. Okay, non-fermenters can be more resistant to antimicrobial agents compared to Enterobacteriaceae. Okay, it's important, if it's available in your laboratory, then it's best to perform a molecular test or DNA sequencing test to confirm for the presence of non-fermenters. Okay, so the most common isolate in specimen from patients with cystic fibrosis is, of course, Pseudomonas aeruginosa. Okay. So the next group for today's discussion is Vibrio, Campylobacter, Helicobacter, Aeromonas and Plesiomonas. Okay. Okay, so for the learning objectives, At the end of this unit, the student should be able to characterize the significant organisms under Vibrio, Campylobacter, Helicobacter, Aeromonas and Plesiomonas, describe and explain the clinical significance and pathogenesis of these organisms, and to distinguish features of the organisms mentioned above for accurate laboratory diagnosis. Okay, so first set we have Vibrios. Okay, naman. Okay, so Vibrios are natural habitants of seawater. So they are mostly halophilic with the exception of cholerae and mimicus. They are not halophilic. They are able to tolerate salt, no, high salt concentration. So they can be called halotolerant. Okay. They can cause infections by drinking contaminated water or eating, or consuming contaminated seafood. Okay. It's isolated mostly from gastrointestinal tract, for patients with gastritis. It can also be isolated from blood and wound infections. The organisms are motile, by virtue of a single polar flagella or flagellum. Okay, they're gram-negative rods, comma-shaped or curved rods. However, in prolonged culture, they may lose that bended shape, okay, and they will retain a straight morphology. Okay, so they could be facultative aerobes, facultative anaerobes rather, or aerobic organisms when cultured. Okay, they are of course oxidase positive except Mechnicoviae, okay, which is oxidase negative, and they are fermentative. No, when it comes to carbohydrate metabolism, they are fermenters. Okay. So culture media that we use for isolating Vibrios include an enrichment media like alkaline peptone water. Okay, it usually has a pH of about 8.4. So initial isolation, for initial isolation, we use enrichment broth, alkaline peptone water. So we inoculate the organisms there, including the swab. Okay, so if you're going to use the swab is included, okay, inside the tube. Okay, so we incubate it for 5 to 8 hours at 35 degrees Celsius. And then afterwards, we can subculture it to TCBS. Okay, so the selective media, that we use for cultivating Vibrios is TCBS, or thiosulfate citrate bile salt sucrose agar. Okay, it contains sucrose and oxgall. Oxgall is a derivative of bile salt. Okay, it also contains sodium citrate and indicators like bromothymol blue and thymol blue. Okay, so the pH is around 8.6. Okay, so the high pH will enhance growth of Vibrios. They cannot be grown in an environment that's acidic. They are killed by acids. Okay, so the high pH, the oxgall, the citrate are important in inhibiting gram-positive bacteria and lactose-fermenting gram-negative bacteria. Okay, sucrose, of course, is added to show or demonstrate sucrose fermentation by the organism. Okay, and if they are able to ferment sucrose, they will produce smooth yellow colonies on the green TCBS. Okay. So this is a table here showing the different species of Vibrio, okay, those that are sucrose positive and those that are not able to ferment sucrose. Okay. So first species, so we'll only be discussing the clinically significant species for this group. We have Vibrio cholerae, the representative organism. It is the causative agent of cholera. Okay, so cholera is a gastrointestinal tract infection characterized by severe diarrhea. Okay, death may occur as a result of severe dehydration. Okay, so Mode of transmission, of course, ingestion of contaminated seafoods or undercooked shellfish or drinking contaminated water. Okay. Vibrio cholerae, again, is non-halophilic, but it's halotolerant and it can ferment sucrose. Okay. So antigenic structure and biologic classification, it possesses a single heat labile flagellar antigen. So H is the symbol for flagellar antigen. Okay, so they only have one, but it's poorly immunogenic. Okay, they also possess somatic or O antigen, which is serologically specific. No? There are about 130 to 139 serogroups of the O antigen. Okay, so there may be different O antigens, but they all share the same H antigen. They only have one of that. Okay. The O1 strain is the most common cause of epidemics and pandemics of cholera. Okay. While the non-O1, so the rest of the 129 serogroups are only able to produce cholera-like diseases. Okay. So the Vibrio cholerae serogroup O1 has serotype O1 has three serogroups. So meron tayo nung Ogawa, which has been isolated in India, Inaba from the Philippines, and Hikojima from Japan. Okay, so again, there are more than 130 or 139 serogroups based on typing for the O antigen. Okay, so yan. Okay, so the O1 antigen can be categorized into two biotypes. So we have the classical biotype and the L-Tor biotype of the O1, okay, serotype. Okay, so to differentiate them, okay, you can perform RBC hemolysis. Okay, so the classic strain Okay, so the classical strain of O1 is responsible for pandemics in the past. Okay, they are non-hemolytic on blood agar plate, Voges-Prescauer negative, polymyxin B susceptible, and they cannot agglutinate chicken RBCs. Okay, whereas the L-Tor strain is responsible for the recent pandemics. These are more aggressive. No, they are beta hemolytic. They are Voges-Prescauer positive. They are resistant to polymyxin B, no? And they are more resistant to environmental changes, and they can also agglutinate chicken red blood cells. Okay. Now, apart from O1, the L-Tor biotype, the Vibrio cholerae O139, which is probably the last, okay, for the O antigen 139. Okay, this is said to have a similar range of disease with the Vibrio cholerae O1 L-Tor biotype. Now, to differentiate, you can perform antigen detection, so you detect O1 antigen, or detect the polysaccharide capsules. So O1 antigen is positive for, of course, Vibrio cholerae O1, and the polysaccharide capsule is present for O139. Okay, so that's how we differentiate O139 and O1 L-Tor biotype because they are said to be similar. Okay. Oh, let me go back to that pala. Okay, so look, okay, so the classical type is responsible for the first 6 pandemics that we have had, no? 1817, 1829, 1846, 1863, 1881, and 1899. Okay, so nagrabi-grabi diri sa pula. Okay. However, for the 7th pandemic in 1961, that has been caused by the L-Tor type. Okay. So for the pathology and pathogenesis, okay, so our Vibrio cholerae is pathogenic only for humans. Okay, under normal gastric acidity, an individual would, for him to be infected, okay, more than 10 to the 10th power of Vibrio cholerae or more has to be present in water, for infection to occur. Okay, in food, less than, so about less than 10:2 up to 10⁴ power of the organisms in food, okay, is enough to cause infection. So kailangan mas marami sa water, okay, to cause infection. That's under normal gastric acidity. Okay, now if the patient is taking acid suppressors, then a small amount may lead to infections of Vibrio cholerae. Okay. Now, the pathogenesis of the infection is as follows. It can produce severe diarrhea. It can also cause mild infections. Okay. The severe type of infection is due to the presence of a potent enterotoxin called choleragen. Okay. This enterotoxin affects the buccal bowel mucosa, okay, which leads to outpouring of water and electrolytes, hence the characteristic rice watery stool, basa ang tae. No? Yun yung effect ng choleragen. Okay? So this would lead to severe dehydration and severe muscle cramping and anuria. Kasi sige-sige ang pupo, no? Ang bowel movement. Okay? So severe dehydration, of course, may lead to hypovolemic shock and circulatory collapse, and eventually would lead to death. Okay? So in mild infections, individuals may be asymptomatic or the diarrhea may be self-limiting. Okay? So the individual may be a carrier. Okay? So Vibrio cholerae usually produces heat labile enterotoxin with the A and B subunits. So yung type ng enterotoxin niya yung may A and B subunits. Its A subunit causes an increased level of cyclic AMP. Okay, so an increase in cyclic AMP levels usually leads to hypersecretion of water and electrolytes into the lumen. Okay, kaya lumalabas lahat. Okay, the diarrhea usually around 20 to 30 liters of water. Okay, I think this is a day. Okay, so grabe kadaghan, no? Na fluids na lumalabas. So this would lead, of course, to dehydration. Kaya dapat paspas din ang fluid and electrolyte replacement. Dahil pag hindi, it would lead to, of course, shock, no? Acidosis and death. Okay, so the diagnosis for the laboratory portion can be done by the usual, no? So microscopy. Okay, of course, the specimen is stool. No? Observe for the characteristic rice watery stool ng pasyente. Rectal swab may be accepted usually in the acute phase or acute stage. Specimen transport may be done with the use of Carey-Blair. Kung kailangan i-transport, then we use Carey-Blair. Okay? So gram staining would result to gram-negative comma-shaped rods. Okay? Motility may be best observed using dark field or phase contrast microscopes. Okay? For culture, we can grow them on peptone agar. No? They can grow rapidly on peptone agar. And then we can also grow them on blood agar plate with a pH 9. And then TCBS, of course. No? So on TCBS, they grow smooth yellow colonies. Okay? Yung ating ito, like this. Okay? Smooth yellow colonies. Okay? Now, to identify, we can also perform string test using 0.5% sodium deoxycholate. And this is added to the organism. Okay, mix it and then observe for the production of a viscous string, this one. Okay, so this one is an indication of Vibrio cholerae presence. And then, of course, serotyping should be done to differentiate serogroups 01 and 0139, especially during times of cholera epidemics. And we can also perform and observe biochemical reaction patterns. Okay, now for treatment, water and electrolyte replacement is really, really important. No? Oral tetracycline may be given. However, note that some Vibrio cholerae may have already acquired assistance because of transmissible plasmids, no? Yung mga jumping genes na yan. And of course, again, perform sensitivity testing or susceptibility testing. Okay, so this table here shows you the different species of Vibrio. Okay, and the tests that you can use to differentiate them. Okay, so Vibrio cholerae is non-halophilic, like Mimicus. For sucrose fermentation, cholerae is positive. Okay, Mimicus is negative. For ADH, positive lang si Fluvialis and Furnacii. Okay, for Cellubios vulnificus, for Voges prescauer, Anginolyticus. Okay. So Vibrio cholerae is the agent of epidemic and pandemic cholera. Now the so-called Asiatic cholera, which was first noted in India. Okay, because of their culture and practices. Okay. And then note, no, Vibrio parahemolyticus, which is the most common agent of infections caused by eating contaminated seafoods like oysters. No? So it is able to demonstrate a phenomenon wherein the strains isolated from human hosts or the clinical isolates demonstrate hemolytic capacities on blood agar plate, okay? While those isolated from non-human sources are not hemolytic, okay? This is observable in a special culture media called the Wagatsuma agar. Okay, so the phenomenon described is Kanagawa phenomenon. This is possible because of the presence of a thermostable hemolysin called the Kanagawa toxin, which is seen only in human samples. Okay, so Kanagawa phenomenon is demonstrated by Vibrio parahemolyticus from specimen isolated from humans. Okay, and this can be demonstrated best in the Wagatsuma agar. The Wagatsuma agar contains, it's a blood agar plate, no, Wagatsuma blood agar plate. It contains high salt mannitol concentration. Okay, so it has mannitol and high salt concentration. Okay, let's move on to Aeromonas. Okay, so Aeromonas is found in both fresh water and seawater. It can cause gastroenteritis, cellulitis, wound infections in humans, no? After coming into contact with contaminated water, either from the drain, the sink, or tap water, and even sometimes distilled water. Okay, but they are also known to cause diseases in cold-blooded animals. Yeah, merong Aeromonas infection sa fish. Okay, lab diagnosis. We can grow them on MAC, on EMB, on SSA. No, so these are same culture media that we use for identification of enteric organisms. So we have to perform oxidase tests. No? We can also grow them on specialized media like the SYN medium, no, remembers? Cefsoludin, Irgasan, Novobiocin medium. Okay, so we can also perform fermentation of glucose and indole production, okay, to differentiate them from Pseudomonas. Okay, they are motile by virtue of a single polar flagella. Okay, the most common human isolate is Aeromonas hydrophilia. Okay, so hydrophilia means water loving. Okay. Okay, so descriptions or characteristics rather, they're able to produce heat-labile enterotoxin and heat-stable cytotoxic enterotoxin. Okay, so may heat-labile enterotoxin at meron din silang heat-stable cytotoxic enterotoxin. They are also able to produce enzymes like protease, lipase, and nuclease. Okay. Then we have Plesiomonas. No? So microscopically, they're motile, straight, ends are rounded. No? Of course, you have negative bacilli. Previously, they belong to the Vibrionaceae family together with Aeromonas. But currently, they are categorized as enteric bacteria. Hence, during your discussion, kasama na si Plesiomonas. Okay, so there's only one clinically significant species for this, and that's Shigeloides. It's important clinically in Japan, and in tropical and subtropical areas also as agent of gastroenteritis. The infection caused by this organism is mild with no blood or mucus in stool. Of course, again, paano nakukuha? Ingestion lang ng maduming pagkain. So ingat-ingat. Ito talaga ang nagdadala ng problema, yung maduming pagkain. Okay? So lab diagnosis, we can grow them on blood agar plate. We can also grow them on MAC and EMB. Of course, they're non-lactose fermenters on MAC and EMB. Okay, they are oxidase and indole positive, to differentiate them from other species of enteric organism. Ayan. So this table shows you the differences between the three members, previous members of Vibrionaceae. So again, as mentioned, this is currently a member already of the enteric group. Okay, so let's move on to the next organism. We have Campylobacter. Okay, so Campylobacter jejuni and Campylobacter cholerae are most frequent isolated species of Campylobacter in humans. Now, technically, they are clinically indistinguishable. Both of them are curved, gram-negative bacilli. But for better differentiation, okay, it is said that it is best to substitute carbofluoxin as the counter stain when you perform gram staining. So remember gram staining, the counter stain uses safranin. However, when your organism is Campylobacter, the differentiation is improved when you change safranin into carbofluoxin. Okay, so there are 19 species of Campylobacter, but the most clinically significant is jejuni, which is the agent of most human infections, followed by Campylobacter cholerae, which causes about 5 to 10% of infections in humans. Okay, so again, they are both microaerophilic, capnophilic, catalase and oxidase positive. Both are non-oxidizers. Okay, they cannot oxidize or ferment carbohydrates. Okay, so green yung dalawang nilang OF media. Okay, they use amino acids as their source of energy. Okay, so they're gram-negative rods. They may show comma-shaped or S-shaped or gull-winged shapes. Mukha daw seagull like this. Ito yung wings ng seagull. At ito naman yung organism. Ayan, so gull-winged shapes. They are able to cause gastroenteritis and diarrhea. And they have been associated in the Guillain-Barré syndrome. Guillain-Barré syndrome. It is an autoimmune disorder resulting from cross-reactivity of Campylobacter antibodies with the nerve ganglia. Okay, so ang mode of transmission is ingestion again of contaminated materials and direct contact. Okay, so direct contact with contaminated animal feces or direct contact with infected person. Okay, animal carriers for Campylobacter are the usual farm animals. Okay, It has a characteristic darting motility, parang mabilis yung movement. No? This particular motility is best demonstrated when using dark field microscope. And so they are motile with single polar flagellum. Okay. So pathogenesis in ang ating Campylobacter. Okay. So it is able to produce cytotoxins, cytotoxic factors, and enterotoxin. The organism is believed to multiply in the small intestine, and then from there they could invade the epithelium, which would lead to inflammation. Eventually, this results in the appearance of blood cells in stool. Okay. Sometimes the bloodstream is invaded and it could cause endocarditis, septic arthritis, and even meningitis. Okay. So to diagnose the presence of Campylobacter, stool would be the best sample. Okay. Of course, gram stains may show gull wing shaped rods that are gram negative. For cultivation, it is best to use selective media. So we have Campylobacter blood agar. Okay, so this blood agar is selective due to the presence of vancomycin, polymyxin B, cephalothrin, trimethoprim, and amphotericin B. Skyros medium is quite similar, but it only contains vancomycin, polymyxin B, and trimethoprim. Okay, the organism is quite difficult to isolate because it needs special conditions for growth. No, medyo OA ito, fastidious. It is incubated at reduced atmospheric oxygen. Okay, so about 5% oxygen lang, with 10% carbon dioxide and 85% nitrogen. The optimum temperature for growth is usually at 42 to 43 degrees Celsius. Okay, so this temperature usually prevents growth of enteric organisms. Patay ng enteric organisms sa 42 to 42 degrees. Okay, growth is usually observed within 48 hours. Okay, the colonies may be gray to pink or yellow to gray colonies. They are, of course, non-hemolytic, and it may be slightly mucoid. Okay, so these are the characteristic species, no, jejuni, coli, and fetus, pero not as common as the first two. Okay, so at 25 degrees Celsius, fetus shows growth. At 37 degrees Celsius, all three species are positive. At 42 degrees, only jejuni and coli can grow. However, to differentiate jejuni and coli, you can perform hypurate hydrolysis. Jejuni is positive for hypurate hydrolysis. For nalidixic acid, okay, both jejuni and coli are susceptible. And for cephalothrin, both jejuni and coli are resistant. Okay. Next, we have Helicobacter organism. So previously, they belong to Campylobacter. So previous name is Campylobacter pylori. So there's only one clinically significant species for this, and that's Helicobacter pylori. The characteristic is that it's small, no, mahirap hanapin microscopically, true, no, curved gram-negative bacilli. Sometimes they only demonstrate the U form. It's microaerophilic, like Campylobacter. Okay, it would require 5% oxygen with 10% carbon dioxide and 85% nitrogen, and it should be incubated at 35 to 37 degrees. It cannot be grown at 42 degrees Celsius. Initra. Okay, it's motile by virtue of a tuft of flagella at the end. Ayan, Solophotricus ito, si Helicobacter. Okay. It's a very, very strong producer of urease. Okay? So it's oxidase and catalase positive, but it cannot reduce nitrate to nitrite. Okay, so for pathogenesis ng ating Helicobacter pylori, okay, so the natural habitat of our organism is the mucus-secreting cells of the stomach. Okay. This organism is known to be the agent of antral gastritis, duodenal ulcers, peptic ulcers, gastric ulcers, and it has also been implicated in gastric adenocarcinoma. Okay, this organism grows best at pH 6 to 7. It cannot actually survive the acidic pH of the gastric lumen, so it burrows itself into the folds of the stomach wall. Remember, yung inner wall ng stomach natin has folds. It hides itself there. Okay, so it invades the mucosa. Okay, and then it releases the toxins and lipopolysaccharides. It damages the mucosal cells. Okay? So it produces proteases and ureases and cause damage to the mucus secreting cells. So when these cells are damaged, they can no longer produce mucus. Okay? Remember, mucus is very important, covering of this epithelium to protect it from the effects of the acids found inside the lumen. Okay. Now, if there are damaged mucus secreting cells, okay, the amount of mucus covering the lining might lessen, and this may cause the acids to damage the mucus cells further. Okay, producing lesions or ulcers. Okay. The presence of the large amounts of urease will produce large amounts of ammonia, and this is also detrimental to the naturally acidic environment. So the large amounts of ammonia produced from the activity of bacterial urease, it will neutralize the hydrochloric acid, which is bad for the functioning of our stomach. Okay. So clinical syndromes. Okay. Again, it is implicated usually in chronic gastritis, in duodenal ulcers, peptic ulcers. Ayan, so nagbubutas-butas. Okay. Normally, the lining of the stomach is wrapped in mucus, okay, due to the mucus released by the mucus-secreting glands, the mucus-secreting cells. However, if the mucus-secreting cells are damaged because of the action and activity of H. pylori, okay, the mucus protecting that wall will lessen, okay, causing the acids to penetrate into the lining, producing these ulcerations. Okay. So gastric biopsy for histologic examination would be best to identify Helicobacter pylori. Okay. So for a laboratory identification of H. pylori, we can perform special silver stain or modified GEMSA. Okay. The gold standard is still histological staining, okay, and of course culture of biopsied tissue from the stomach or the duodenum. Okay, so what do we do with the tissue? We mash it or ground it, and then take a sample of that mashed tissue and plate it on ship's blood agar, incubate it at 37 degrees Celsius under microaerophilic conditions and increased humidity. Eventually, colonies would grow, showing small, gray, translucent characteristics that are slightly beta hemolytic. Okay. Biochemical testing can also be done. Okay. Remember, they are able to strongly produce and rapidly react, producing the positive color reaction that we see in biochemical testing. Okay, so they are strong producers of urease. Okay, so diagnosis, apart from cultivation, biochemical test, culture, we can also identify them using serologic methods. We can determine serum IgG antibodies for H. pylori. We can perform stool antigen tests via ELISA. We can also perform carbon urea breath test, which detects urease activity. Okay, so this is usually done to confirm Helicobacter colonization and to monitor the treatment for H. pylori. Okay, so breath test is commonly done for that. Now, endoscopy is also done to in gathering samples, for performing biopsy, collecting specimen for testing, and microbial culture. Okay, so for treatment, like those previously mentioned, it's best to perform or to give triple therapy, metronidazole or bismuth subsalicylate or bismuth subcitrate plus amoxicillin or tetracycline. So tatlo, metronidazole, and then a choice between bismuth subsalicylate or bismuth subcitrate plus amoxicillin or tetracycline. So that treatment is given for 2 weeks. Okay, so aside from that, we can provide acid suppressing agents and proton pump inhibitor. Both will lessen the acidity of the environment and help the ulcers heal. Okay, so this is usually given for 4 to 6 weeks. Okay, so this is a table showing the differences between the species of Campylobacter and the Helicobacter species pylori. Okay, so to differentiate, jejuni and cholerae perform high purate hydrolysis, positive si jejuni, negative si cholerae. Okay, what else? Fetus is positive for growth at 25 degrees Celsius and susceptible to cephalotin. Helicobacter pylori is easily distinguishable by a strongly positive production of urease enzyme, and it is resistant to nalidixic acid. Okay, so key points before we end this discussion. So the key points is that most species in this group are found in fresh or estuarine or marine waters. Vibrio cholerae produces a powerful enterotoxin, choleragen, and is responsible for large numbers of epidemics and pandemics. Okay? Other Vibrio species are common causes of diarrheal diseases, usually related to consumption of raw shellfish or related to aquatic wound infections. Okay? TCBS, of course, is the agar or the medium of choice to isolate and differentiate Vibrio species. Okay? Aeromonas species can cause infections that may lead to septicemia, meningitis, and even death. Okay? So, Campylobacter naman are generally responsible for enteritis. So, jejuni is one of the most common causes of bacterial diarrhea worldwide. It has a characteristic seagull wing shape or morphology under the microscope. And it is associated usually with the infection or with the syndrome Guillain-Barré syndrome. Okay, so patients that are positive with this syndrome are almost always positive for Campylobacter antibodies. Okay? And then H. pylori is strongly associated with gastric and duodenal ulcers. So it is able to cause the production of such infections due to its activity. It is able to produce urease, which produces ammonia, which affects the acidity of the gastric environment. Okay? And it has also been implicated in gastric cancers or gastric carcinoma. Okay, so that's the end of today's discussion. Thank you for listening.